Okay, let's go in and get started for today. So remember that you have the problem set that's going to be due on Wednesday, so everything's been covered for that. So you should be fine. In terms of quiz 10, it's currently set to be due on Wednesday. There is a question on that regarding ATP synthase, which we haven't even touched on yet. So I'm going to go ahead and, well, I'm going to ask you to hold off on that one. We'll see where we get on Wednesday. And then for now, I'll just keep the due date as Wednesday, but we might not get to that material and then we'll have to delay it again. So, okay. Again, of course, remember the exam has been delayed 2 days. The 17th is when we'll have the exam, okay? All right, so our riddle, I'm where yesterday follows today and tomorrow's in the middle. What is it? Day after tomorrow? No, that's not it. What's that? No? No? Anyone? Huh? No, not midnight. It's a dictionary. Right? Dictionary. Yesterday follows today. Okay. And tomorrow's in the middle. In a dictionary. If you look them up. Some of you are starting to get it. Some of you are saying it's too nice to be outside or to not be outside and in here. So listening to your stupid riddles. Okay, anyway. You don't have to get mad at me, okay? All right. All right, so Friday, we left off right here, really. I said we'll cover this today, of course, on Monday. We're talking about this really important topic, oxidative stress. It's important because of its prevalence in terms of possibly the causative agents of many diseases, many really awful diseases that end up killing us. Okay, so very important subject, and we put it here. I put it here in this this lecture because oxidative stress can increase or it can decrease based off of what's going on in the electron transport chain. So essentially, if there's not a proper balance of the number of electrons going into the electron transport chain versus the ones that are coming out, that can lead to increased levels of oxidative stress. Okay? Of course, electrons enter into the chain primarily by NADH. That's what most of the redox reactions in the cell create is NADH. But of course, FADH will feed into it as well in a variety of places, leading to the reduction of ubiquinone, making ubiquinol. That's how electrons get into the chain. The only way that electrons get out of the chain is through oxygen. So you have to have, first of all, a supply of oxygen to get those electrons out, and then you have to have a proper flux through the transport chain for there to be this continual turnover of electrons going on to oxygen. If that flux gets out of balance, then you can get this accumulation of electrons in the chain, and that can then lead to the production of reactive oxygen species. So we actually looked at this, these reactive oxygen species on Friday, and we looked at how the mitochondria will deal with them and take this superoxide radical using this enzyme superoxide dismutase and make hydrogen peroxide from that. Glutathione peroxidase can then reduce that hydrogen peroxide to water, but it has to have an electron donor in glutathione to do that. Okay. Glutathione, once it's used in this reaction, becomes the oxidized form of glutathione, and that has to be re-reduced by glutathione reductase, which uses NADPH to do that. The source of NADPH in the mitochondria is this is made by this enzyme nicotinamide nucleotide transhydrogenase, which takes electrons off of NADH, which if electron transport chain is slowed down, will start to accumulate in the matrix of mitochondria. And it will put those electrons in NAD+, making NADPH. Okay? Now that's how, by and large, mitochondrial metabolism will deal with the production of reactive oxygen species. If we were to move to the cytosol, okay, and oxidative stress can be a problem in both the cytosol and the mitochondria. In fact, mitochondrial oxidative stress can lead to increased oxidative stress in the cytosol. Right. There has to be some way then in the cytosol for the oxidative stress to be managed as well. And what you are looking at is a pathway that will allow cytosolic environment to handle or manage increases in oxidative stress. Okay. The pathway is actually referred to as the pentose phosphate pathway. I'll write that down. Okay. It is a classic example of what would be considered a carbohydrate metabolic pathway. Okay. And oftentimes it's just commonly referred to as PPP, okay, pentose phosphate pathway. Pentose meaning five carbon sugars in particular. Okay. I didn't cover this in carbohydrate metabolism. I held off to cover it because of its connection with oxidative stress, though it has multiple functions, this pathway. And I'll mention some of those functions beyond just its connection to oxidative stress, though we'll, of course, talk about what it does in terms of oxidative stress as well. Okay. I'm not going to go through the pathway in a step-by-step method and all of the enzymes in it. I will mention a couple of the enzymes, and then we'll just leave the rest to your imagination or your own research. Okay. But the pathway can be split into two really phases. Okay. And a cell doesn't have to run both phases. It can run either one or the other. Okay, but one of the phases would be considered the oxidative phase. That's really what we'll be concentrating on. That would be everything to the right of that line that I just drew. Okay, the non-oxidative phase is important in many cell types. I'll mention it, but I'm not going to go into it in a lot of detail. Okay. Now, the pathway itself, the pentose phosphate pathway, starts at a hexose. Okay, glucose 6-phosphate is where it actually starts. Of course, glucose 6-phosphate is the product of the trapping reaction. Okay, hexokinase. The beginning of course, glycolysis as well. Glucose 6-phosphate has a number of fates in the cell, and one of them is shown here. There are two enzymatic steps here, number 1 and number 2, recognized by that small little arrow. But two steps will essentially oxidize glucose 6-phosphate to 6-phosphogluconate. Okay. Now, here's where we want to start making our connections to this. The enzyme that's implied by this arrow is something called glucose 6-phosphate dehydrogenase. Okay. Glucose 6-phosphate dehydrogenase, sometimes it will just go by G6P dehydrogenase. Okay. I want to mention that enzyme because you'll notice that it uses a mole of the NADP plus and makes NADPH in this reaction. Okay? I'm going to put a star by that NADPH. Okay? Now, let me go to the next step in the pathway. The other one I want to highlight here. This is the third step in the pathway. Okay? 6-phosphogluconate, it undergoes an oxidative decarboxylation reaction to ribulose 5-phosphate. Okay? The enzyme there is something that we call 6-phosphogluconate dehydrogenase. That's fun to say, that's fun to spell. 6-phosphogluconate. That enzyme also uses a mole of NADP plus and will also then make NADPH. Okay. Now the rate limiting step of this pathway, okay, irreversible, is catalyzed by glucose 6-phosphate dehydrogenase. Now, in the first 3 steps of this pathway, you'll notice then that 2 moles of NADPH are made. What's the importance of that? Well, if we go back here, okay, we talked about in the mitochondria how superoxide dismutase can make hydrogen peroxide, and then you have glutathione peroxidase that can make water from that, and then you have to have NADPH to re-oxidize glutathione using glutathione reductase. That exact same process occurs in the cytosol as well. Okay, so if oxidative stress goes up in the cytosol, there is a cytosolic version of superoxide dismutase that will make hydrogen peroxide. Glutathione peroxidase will use NADPH to then make water, and of course it uses glutathione, reduced glutathione, and then there has to be something present to re-oxidize glutathione, okay, which NADPH is going to be doing that used by glutathione reductase. Okay. So the pathways in the mitochondria and the cytosol are really very similar in terms of how they deal with the reactive oxygen species. Their source of NADPH, though, is different. Okay. The pentose phosphate pathway provides the NADPH, quite a bit of it actually, for the purposes of re-oxidizing glutathione to the reduced form of it. Okay, and that's actually shown here. Okay, glutathione reductase. Now don't think, oh, it's only the NADPH that's produced by the first step that's used by glutathione reductase. Don't think that. Okay, it's just shown there. NADPH, generally speaking, is going to be used by glutathione reductase. Okay. Now, a couple of other things to say here. The NADPH that's made by the pentose phosphate pathway, while being very important to diminish the effects of oxidative stress, that NADPH, and they show this here as well, okay, is also very important in reductive biosynthesis. Okay? Very important in particular in actively dividing tissues. Okay, that are going to need to carry out lots of biogenesis of, let's say, lipids or nucleotides or amino acids even. Okay, those pathways, if you really dig into the details of those pathways, in particular fatty acid and lipid production, require a significant amount of NADPH. Okay, so this pathway will increase in flux if the cell is undergoing significant biogenesis. Okay, to produce the needed NADPH. In fact, if you look down here, something that we didn't cover, this would be steps 4 and 5 of the pathway. Well, really step 4 is what I'll highlight. Okay, step 4 can take that ribulose 5-phosphate and make ribose 5-phosphate from it. Okay, this would be multiple steps, but that ribose 5-phosphate can then be used in nucleotide and coenzyme synthesis, along with the NADPH that's made. Okay, so this is a really important pathway for a number of reasons. Certainly the connection to oxidative stress, okay, and then of course the connections to biogenesis pathways, lipids, and of course nucleotides. Nucleotides being the production of ribose 5-phosphate. This is the sugar of the nucleotide. Okay, and of course NADPH. Now, the final thing here, and I said I mentioned it very briefly, but there can be two fates of ribulose 5-phosphate. Ribulose 5-phosphate can just be used to regenerate glucose 6-phosphate using these enzymes trans-ketolase and trans-aldolase. Multiple steps. The stoichiometry is kind of a headache when you start looking at it. I'm not going to go over it in any detail. But ribulose 5-phosphate can just refeed glucose 6-phosphate as well. Okay. In that case, you'd have a cell that really doesn't need ribose 5-phosphate production, and it's really just needing the NADPH. Red blood cells would be a really great example of this. Red blood cells don't really need nucleotides. Of course, they don't have a nucleus. They're terminally differentiated. They are not going to divide any longer, so they don't really need that much nucleotide synthesis. And so they'll just recycle the ribulose 5-phosphate back to glucose 6-phosphate. They need quite a bit of NADPH, though, because there can be significant oxidative stress in red blood cells. Okay, so that's the pentose phosphate pathway. That's truly just a quote-unquote crash course on the pentose phosphate pathway. Lots of enzymes, lots of other things that we could go over, lots of details there. It's a very important pathway currently in metabolic literature because of its connections to lots of different things. But we're not going to go over more. We're not going to go over this in more detail than that. Questions on that? Okay. I will say last thing here, just in terms of primary literature and that kind of thing. This pathway, as you might expect, because of its connections to biogenesis, is found upregulated in cancer metabolism as well. Okay. Not surprisingly. Okay. All right, now I've got a question for you. Okay. Let me come here. Let me get back to that here in a little bit. Okay. All right, so this is a big one. Let me go ahead and get this a full screen. Pause that. Okay. So a family on vacation in Greece sit down to an authentic Greek meal complete with falafel. And you think, no, it sounds yummy. Okay, falafel, the main ingredient of falafel is fava beans, if you're interested. Okay, just so you know. It's delicious. All right, anyway, a day later, one of their children's skin has a yellowish tint and is clearly in need of medical attention. In the ER, the physician determines that the child's red blood cell count is low and their kidneys are beginning to fail. Upon learning of the child's diet in the past 24 hours, the head physician orders a genetic screen. What gene do you think is the target of that screen? And you think to yourself, interesting. Fascinating question. I'll give you a minute 41 to talk about that with your neighbor. What do you think? Just give me a gene, and we'll go from there. Your marks, get set, go. Minute 41, what is the target of that gene? About 30 seconds left, so submit an answer. Okay, about about 5 seconds left. So submit an answer, even if it's a single letter. Get credit for being here. Okay. Awesome. All right, let's check this out and see what people said here. That's fine. Show results. Okay. Red blood cell gene. Very good. A red blood cell one. HIF1. Oh, it's interesting. We'll talk about that. A kidney gene. Glucose 6-phosphate dehydrogenase. Glutathione dehydrogenase. CREB. Yes, because CREB. So, you know, if you're taking BC463 right now, maybe CREB is just like, it's the everything gene, right? I mean, it just does everything, and so I'll just say CREB, and we'll just go from there. Okay, you tell me. Now, that's what I was looking for. So, good effort. Okay, anyway, let's highlight this. So, glucose 6-phosphate dehydrogenase. That's the right answer. Okay, so let me go ahead and kind of take you through this just from a symptom standpoint, and then we'll talk about why the genetic screen. Okay, again, this is on this special topic, oxidative stress. These symptoms are indicative of someone that is undergoing hemolysis. Okay, red blood cell count is low, which would seem to indicate that something's going wrong in the blood. Potentially they're losing red blood cells. Okay, now the jaundice, the yellowish tint on their skin, that will be due to the fact that if red blood cells are being lysed, hemoglobin's being dumped into the plasma, it will be absorbed by the liver. The liver will metabolize heme. It'll have an abnormal amount of heme metabolism, and one of the breakdown or byproducts of that pathway is bilirubin. And bilirubin will then be released into the plasma, and that can actually cause this yellowish kind of tint, even in the eyes, okay? And that would definitely be indicative of fairly massive amount of red blood cells being lost, okay? Now, so what's the deal, okay? And what's the deal? What's the deal with fava beans or falafel? Why is that a concern? And why glucose 6-phosphate dehydrogenase? Well, let me go here, okay? Come back here. We've talked about this, okay? The last several days, we talked about this happening, right? The production of superoxide radical, conversion to hydrogen peroxide. Hopefully, glutathione peroxidase is able to make water from that. So we talked about that enzyme. Of course, we need the glutathione, but of course, to reoxidize glutathione, we need glutathione reductase, and to do that, we need a source of NADPH. Okay. And one of the sources of NADPH in a cytosolic environment, okay, would be glucose 6-phosphate dehydrogenase. Okay. Now, in a red blood cell, understand there's no mitochondria. Okay, so you really only have one available resource for NADPH, and that's the pentose phosphate pathway. So if this enzyme is not functioning, then you won't get this, and you won't get this, and you won't get this. And what will happen then is that this will be the outcome, right? Production of a hydroxy-free radical, and then you'll get oxidative damage to, in particular, in this case, lipids. Okay, the lipids will undergo peroxidation. That will disrupt membrane integrity of the red blood cells. They'll start to lyse. Hemoglobin will go into the blood. Bilirubin is the result of that. Okay, so one of the potential candidates for these types of, or for these symptoms, would be an acute increase in oxidative stress. Okay. Now, what's the deal with fava beans? Well, fava beans, okay, or falafel produce this. Okay, one of the compounds in that bean is adenosine, and that will lead to increases in oxidative stress. Now, an individual with healthy glucose 6-phosphate dehydrogenase can have falafel and have no problems. The rest of the family, except for this poor kid in Greece. Okay, all right. But there's a significant portion of our population that has a genetic disorder in glucose 6-phosphate dehydrogenase. It's actually called glucose 6-phosphate dehydrogenase deficiency. Okay. And by significant, 400 million people suffer from this. So it's a lot. It's not a rare genetic condition. It's pretty common, actually. You can go your entire life and not even know you have it, as long as you don't have an increased incident of oxidative stress for some reason. Okay, so a child could go asymptomatic for years. No one would know the difference until they were put under conditions where all of a sudden oxidative stress were spiked. Okay, falafel, the kid must have porked out on falafel, right? I mean, they were like, oh yes, I love this. Oh. Right? That's how kids are. I've seen it. Like, don't eat anything, don't eat anything, don't eat anything, and then I find something I like and they just eat all of it. Okay? So that could have been what would happen here. Okay? Obviously, this is a hypothetical scenario, but it has some bearing in reality. Okay? So that then, right, so then from a physician standpoint, you can order a genetic screen and find out, would this individual have glucose 6-phosphate dehydrogenase deficiency? And then that would allow them to understand better how to treat that individual. So that would have been the genetic screen. Yes, question?
So is it like somewhat common that people eat falafels or similar ingredients and then have an issue like this? I mean, it's like a fairly...
Yeah, probably not one meal.
Situation.
Yeah, but okay, the question would this be a common situation if people eat falafel? Probably not one meal, okay? I mean, a child maybe, but there actually is a condition known as favism, okay? So people that, especially of Mediterranean origin, because it's a pretty common ingredient in a lot of their foods, people will eat a lot of that, and as it starts to accumulate in their bodies, it can lead to increased incidence of oxidative stress, and they can get what's called favism. Favism is really caused by this, glucose-6-phosphate dehydrogenase deficiency. Okay, it was called favism at one time before really modern medicine, was able to diagnose what was going on, but it was linked to, at some point in time, linked to fava beans. Okay. Yeah.
So with somebody who is suffering from this, usually how they fix it? How they help the person?
Okay, so the question is, how do they fix this, or what would be the treatment of this? So avoid fava beans, all right? I mean, no, but there's not a cure to it because it's a genetic disorder. Okay, so you would have to take increased amounts of oxidative or antioxidants. Okay, that would be the really best way to do it, and also avoid situations where oxidative stress could go up. But I'm not sure of like specific, treatments of it. So, but that would be my best guess. Like I say, you can go. I mean, a person can go asymptomatic. I mean, not show any symptoms. So in some cases, it's just not bad. I would imagine there's varying degrees of how severe it is, depending on how much activity of this enzyme they've lost. Okay, if it's completely dead, then that's going to be a more severe case. So now here's what's another interesting little factoid about this. This disorder, okay, is found, or there's a higher incidence of this disorder in tropical areas. Okay, India, Africa, the Middle East. The Middle East is not really tropical. Okay, but in places where mosquitoes carry malaria, and the reason is because malaria has a really complicated life cycle. A portion of that life cycle resides in your red blood cells. And malaria is really sensitive in terms of its life cycle to oxidative stress. If you have increased incidence of oxidative stress, it will kill the malaria parasite. And so people that have this disorder have a selective advantage over people that don't, because they actually are more resistant to malaria, and they will then of course make it to reproductive ages and pass on that gene, okay, to their offspring. So you'll find an increased incidence of this disorder in those areas where mosquitoes carry malaria. Okay. In fact, if you look at this slide, you have anti-malarials. Primaquine actually increases oxidative stress. Is not good for you unless you have malaria. And then of course malaria is really deadly. It can kill you. And so one of the defenses against malaria is to take primaquine, and that will then increase oxidative stress in your red blood cells. So fascinating stuff. Isn't that fascinating stuff? Questions on this? Okay. So just watch what you're eating. When you eat falafel, just, look in the mirror every once in a while. Am I yellow? Do I feel weird? Yeah, question.
I might have missed it, but why are the kidneys failing?
I didn't say, and I don't know. Yeah, so, I'm sure there's a really good reason, and it's probably really easy to understand, but I don't know. I didn't look into it. Yeah. So, okay. Other questions? Okay. All right, let's do this. I asked that question. We've gone through that. One or two more slides on this topic of oxidative stress. Like I said, it's really important, so I wanted to spend some time on it. And oxidative stress, interestingly and almost paradoxically, can be the result of hypoxia. Okay, so reduced oxygen. You say that can't be. If you reduce oxygen, certainly you're not going to increase oxidative stress, but it actually is true that you can do that. Okay, and the reason is again, you have an imbalance of the electrons going into the electron transport chain and coming out. Okay, the only way electrons come out of an electron transport chain is through oxygen. So if you have a reduced amount of oxygen, you can have more electrons that are entering the chain than are leaving the chain. And so then you get accumulation of electrons in the chain, and then you get oxidative stress. So hypoxia, strangely enough, can lead to oxidative stress. Okay? So we'll start up here. That, and we won't talk about the signals that will lead to this, but that will lead to an increase. Let me go ahead and focus in on this. An increased genetic expression of a factor called HIF. And HIF, and I saw actually, it was interesting, one of the responses to our question, that last question was a genetic screen for HIF, which actually is logical. But anyway, this would be hypoxia. HIF stands for hypoxia induced factor. HIF. Low levels of oxygen can lead to an increased expression of hypoxia-induced factor. Now that is a transcription factor that will bind to the promoter regions of a large number of cells. Not large number of cells, large number of genes. Okay, and I want to mention a couple of those that have a correlation with what we've talked about in this class. Okay, it won't go this direction, but it will go the other direction. Okay, let's start here. Increased levels of HIF will lead to increased levels of PDH kinase. And all of you say, haha, yes, PDH kinase, that's right. It's before spring break, it seems like. No, it's just after spring break. Okay, so PDH does stand for pyruvate dehydrogenase, and then of course kinase. This is the kinase that will phosphorylate the PDC complex, E1 of the PDC complex, and inhibit it. All right, so what you have, if you increase genetic expression of that, you will inhibit pyruvate dehydrogenase. Now, why would you want to do that? Well, that will prevent conversion of pyruvate to acetyl-CoA. That will slow down citric acid cycle flux, which will then reduce NADH that's entering into, and FADH2 that's entering into the electron transport chain. So by doing that, then you're reducing the number of electrons going in the electron transport chain because they're not leaving. You have a hard time getting them out because you have low oxygen levels. And by doing that, then you'll reduce oxidative stress. Okay, so that's one of the first things that will happen in these cells. Okay, interestingly, just simply reduced materials going into the citric acid cycle. Okay. Now, that's really the connection to oxidative stress. Let me go ahead, though, and mention some of these other things here that you'll notice. You'll also notice that HIF leads to increased expression of glucose transporters, increased expression of many of the glycolytic enzymes, and increased expression of lactate dehydrogenase. Okay. Why is that fascinating? Well, many tumors are hypoxic. They have low oxygen supply because they're not really well vasculated. Right? They don't have the capillary beds that integrate with the tissue, and therefore they get poor oxygen supply. Those tissues then, okay, tumors of course, cancer, will then have HIF increase in terms of genetic expression. That will lead to the expression of things that get glucose into the cell, things that use glucose, and then things that convert glucose to lactate. Remember, the Warburg effect says that cancer cells absorb about 20 times more glucose in your normal cell and convert it to lactate. Well, one of the reasons that's the case is because, and not in all cases, but in some cases, or many cases, is because HIF is active, increasing the expression of all of these genes. Okay. So there's a connection here to not only oxidative stress, but also to cancer. Now, interestingly, in HIF also, and I don't have it on the slide, will increase, and I can't remember the name of the hormone, it'll increase expression of a peptide hormone that will actually be a signal. It can be secreted by the cell to start producing blood vessels that will come and feed oxygen or blood to those tissues. So it's not on this slide, but it's interesting. Okay. Now let's step one step further here in terms of cancer. When you look at this, okay, this is really more cancer than oxidative stress, but there have been and has been, given these things, an attempt, okay, to inhibit everything that I just boxed in there. Those are compounds that are potential anti-cancer therapeutics that are targeting hexokinase, the trapping reaction, and glucose 6-phosphate dehydrogenase. The reason that they're trying to do that is to really increase oxidative stress in those cells that could potentially kill cancer. Problem, of course, with all anti-cancer therapeutics is that it will do that to many of your other healthy cells, and it will make you sick. Okay. But that will be, the theory there is to, of course, decrease levels of glucose 6-phosphate so that then glucose 6-phosphate can't feed into the pentose phosphate pathway. So what you're looking at here, this is the pentose phosphate pathway. And if they're not feeding into the pentose phosphate pathway, then you're not getting NADPH and you're also not getting ribose. Okay, so these cells would not be able to produce as much nucleotides or the other materials that they need to continue to divide. Okay, so lots of interesting connections into not only oxidative stress, but cancer metabolism as well. Questions? Huh? It's nice out there, right? Yeah, I know. Don't worry, this will be a really fast 20 minutes. Okay. And you say, I'm not going to be here for 20 minutes. Well, okay, I'll make it 10, 11, 12 minutes. Maybe 13. We'll see. We'll see how it goes. And we won't talk about the Rockies, you know. Teams continue to score more runs than us. It's a problem. I am telling you, they need to hire me. Okay, I've got it figured out. The Dodgers scored more runs than us over the weekend in all 3 games, and that was bad. So significantly more runs, actually. But I mean, the Dodgers are outscoring everyone, but it hurt me. I mean, I'm okay. It's okay. So I'm fine with it. I have to face the music sometimes as a Rocky fan. Okay, all right. No questions? Awesome. Let's leave oxidative stress, okay? So oxidative stress done. Now we can return back to just a discussion on oxidative phosphorylation, okay? So we have now seen how electrons get through the electron transport chain. We've seen this, okay? And we are now ready to go ahead and talk about this guy all the way at the end, the strange looking thing, FOF1 ATP synthase, okay? And how it's going to use protons to make ATP. Now, there's a couple of topics before we get into the mechanism of that enzyme. And it's really fascinating. We'll probably get there Wednesday and probably finish it on Friday. Okay? But before we get to the mechanism of that, I want to go ahead and talk about something called PO ratios, and we'll also then talk about uncoupling. Okay? And PO ratios, just so you know, okay, you may have heard this term, even in this class I think I've talked about it, but PO ratios means this: ATP, and box the P here, produced per oxygen atom reduced. O would be oxygen. Okay. Of course, the oxygen atom comes on molecular oxygen. But that's where we get this idea, PO ratios, ATP produced per oxygen atom reduced. Okay, this requires, to reduce an oxygen atom, that requires 2 electrons. And those 2 electrons in electron transport chain or in oxidative phosphorylation can come from either NADH or they can come from, I'll put succinate, because that's what really brings them to the chain, slash FADH2, which really is going to be the thing that accepts them from succinate. So that's a PO ratio. ATP produced per oxygen atom reduced. 2 electrons are required for that. They come from either NADH or succinate. Now I got a question for you. Another question. Oops. There we go. Okay. Oh, multiple choice one. Okay. I know, easy to respond to, all right? Okay. Yeah, I know, you have to get it right now. Okay, so how many ATP can be made from NADH and FADH2 or succinate, respectively? Okay, so the first number on this list is from NADH. Second number on that list is from FADH2. These would be PO ratios. Okay, how many ATP can I get from those two things? Okay, 3.5, 2.5, 3, or 2, 2.5, 1.5, depends on the size of the C ring, none of the above. Okay, I'm going to go ahead and give you a minute 45 to talk to your neighbor about that. What do you think? On your marks, get set, go. What do you think? Okay, you got 45 seconds. Plenty of time. Okay, about 10 seconds left. So submit an answer if you haven't done so. Get the credit for being here today. All right, 2 seconds left. Okay, let's check it out. See what people put here. Okay. 81% of you say 2.5 and 1.5. Okay. 9% of you say 3.5, 2.5, and then 3 and 2 is 7%. And then 3% depends on the size of the C ring. So 3% of you are right. So you people are like, well, yeah, you never said anything about a C ring. What the hell is a C ring anyway? I mean, you can't trick me like that. I know, I did though. I have to do it, okay? So at this point in time, I can see why 81% of you said 2.5 and 1.5, because all of you have had a group work where I told you that 2.5 and 1.5 were the ATP that's gonna be made from NADH and FADH2. So not only did I trick you there, but I'm tricking you here. Okay, I know I'm tricky. It's okay, you're gonna get over it, okay? I had a bad weekend in fantasy baseball too, if you're really concerned about it. But my wife continues to tell me that it's actually not real, but it feels real, okay? So, I mean, one of my strategies is to yell at all my players, but I don't know them. So that's the problem, and they're, you know, anyway, I have no idea how to get a hold of them. So, but that is, I don't know what that has to do with this. It doesn't have anything to do with this, but we can get back to this, okay? So we are going to talk about the C ring, okay? I promise you. We're not going to talk about it here, okay? But the numbers that you have been told, because 3 and 2 is a common number. How many of you have seen 3 and 2 in another class? A couple of you, right? Maybe you put 3 and 2. Okay, so like I think the Life 102 still teaches it 3 and 2. And so there's lots of classes that will teach the PO ratios as 3 and 2 for NADH and FADH2. I said 2.5, 1.5, but it really does depend on this thing called the C ring, which when we get to the mechanism of ATP synthase, I'll be able to show you why it depends on the size of the C ring. Now, let me say this, okay? We'll do this, okay? If, so this is an if the C ring, I'm going to make some of you feel better, 81% of you actually. If the C ring is 9 subunits, it takes 4 protons to make 1 ATP, okay, in OP, in oxidative phosphorylation. Okay, so that's what we're going to say. If the C ring is 9 subunits, it will take 4 protons to make 1 ATP in oxidative phosphorylation. And you say, okay, awesome. So where do I get 2.5 ATP per NADH? Or 1.5 ATP per NADH? Because you said you're going to make 81% of us feel better. Well, let's look up here, okay? NADH moves electrons into complex 1 onto coenzyme Q. We talked about that process. That will lead to the translocation of 4 protons, okay? That's 1 ATP. Because those 4 protons will come back into the mitochondria, the matrix of the mitochondria, and make an ATP. Okay, now keep following those electrons, all the way to complex 3, okay, to cytochrome C. That will pump an additional 4 protons, 1 more ATP. Okay? Because those 4 protons, again, will come back into the matrix. And then finally, those electrons go from cytochrome C, okay, to complex 4, and an additional 2 protons will be pumped to the P side, and that will make half an ATP molecule. And you say, how can you make half an ATP molecule? Statistically speaking, okay. I understand you can't make half an ATP molecule. Right, but that is where we get this number, NADH, right, will equal 2.5 ATP. Okay, that's where that number comes from. Okay. But it does make this assumption that the C ring is 9 subunits. And that's an important assumption that has been implied that you've never even probably, well, been told about that. There's 3% of you that may have heard of that, okay? Okay, or you just said, sounds too random to be not right, okay? Which is not a great way to go about it, you know, answering a test question. But anyway, right? Now, obviously, if you sit here and think about succinate, okay, we can go succinate. Succinate, it's going to do this. It's going to go through complex 2, no protons pumped. Those same electrons make it to cytochrome C. That will pump, right, 4 protons, 1 ATP. Those same electrons will then make it to complex 4, pump an additional 2. That will make 1.5 ATP. So this is equal to 1.5 ATP. So that's where those numbers come from. Okay. Okay. So you may never have the PO ratio explained to you other than just say this is what they equal. That's where these things are coming from. Okay. And again, we'll get to the importance of the C ring later on, hopefully on Wednesday or worst case Friday. Okay. But that then can lead to something like this. I'm not going to spend a lot of time on this because you have already done this in a group work project. Okay. So I'll just reference, you know, that citric acid group work project and say this is what you were supposed to do. You would have come to, based off of PO ratios, okay, this is PO NADH equals 2.5, FADH2 or succinate equals 1.5, okay? And it's going to produce about 30 to 32 ATP. Right? If you were to use 3 and 2, it will be 36 to 38 ATP. Which you may remember, some of you may remember from a, like, you know, lower level biology class. A lot of times you'll say, oh, per glucose molecule you get 36 to 38 ATP. Well, that assumes a PO ratio of 3 to 2. Okay. And the subunits matter there as well. Okay? All right. Now, I don't have any time to go any further, which is, I'm sure you're also sad. Okay, you have to go back outside. So get out of here. I will see you on Wednesday. We'll talk about uncoupling and we'll get into ATP synthase. Okay?